Good morning, students. Welcome to activity module of Cell, the fundamental unit of life. In this module, we are going to have some fun while learning by doing. So let's begin. Now here is a question for you. Did you ever take note of diffusion happening around you? You already know that osmosis and diffusion are two processes that help in movement of substances in and out of the cell. But did you notice that diffusion is also happening around you and several other places? For example, in a hot cup of tea. When you put a tea bag in a hot cup of water, what is happening is the extract of the tea is diffusing out of the tea bag, that is from its higher concentration to its lower concentration, thereby coloring your tea or flavoring your tea. You also see it when you open a cold drink bottle. A chilled cold drink bottle, when you open it, as you can see in the picture, you see the bubbles rising up. It is because the carbonated drinks contain carbon dioxide dissolved in the liquid. As you release the pressure of the cap off, the gas tries to come out from its higher concentration to its lower concentration. You also notice that if you pour this drink into a glass and leave it for some time, the fizz dies down because again the gas has diffused out of it. Now let's look at some more examples. You have also seen essential oils being diffused. Now what you see in the first picture here is a reed diffuser. Reed are these sticks that are put in a bottle containing essential oil or some perfumed liquid. What is happening is these hollow sticks absorb the liquid and then diffuse it out. Since they get concentrated in that liquid or perfumed substance, they diffuse it out slowly into the air, thereby diffusing the perfumed liquid or perfume out into the environment. What you see next on the right side is actually a little experiment that you can perform. What you need in this experiment is a bowl of water, preferably at the room temperature, not chilled, not hot, not warm water, room temperature. Next thing you need is a freshly cut leaf. The leaf can be of any plant, but it has to be freshly cut. What you need to do is dip this leaf into the bowl of water and leave it for some time. You will see after some time that small bubbles appear on this leaf. Now why is this happening? We all know that leaves perform photosynthesis, and during the process of photosynthesis, gaseous exchange happens through the stomatal pores. So as the leaf is freshly picked, when you dip it in water, the bubbles of the gases coming out of the stomatal pores get trapped inside as tiny bubbles that you can see. This also shows that diffusion is happening around you. Have fun with this activity. Please perform it. Moving on to osmosis. Now let's perform the experiment number 1 of osmosis. What you need, the things you need for this potato osmosis experiment is number 1, 2 to 4 slices of potato, preferably equally sized. You have to cut them already and keep them ready. You need about 4 tablespoons of salt. You can also take sugar, but I take salt. Then you need 2 separate bowls of water. The water has to be tap water. And you need some kitchen towels over which you will place these potato slices. What you have to do is cut 2 to 4 equal size potato slices. I've used 4, you can use 2. And immerse 2 of these slices in a very salty solution of water, which will contain about 4 spoons of salt in a cup of water. Make sure the water is equal in both the containers. Then immerse the other 2 slices in simple tap water containing no added salt. And then leave these 2 solutions with potatoes in them for about 20 to 30 minutes. And see what happens next. After 20 to 30 minutes, remove the potato slices on the kitchen towels as you can see and observe the difference between them. Now since we put salt in one of the solution, the one in the salty solutions will have become soft because of exosmosis. So when you try to tear this potato, which was dipped in salty solution, it will tear easily because it has become limp, because the cells in the potato have started to shrink because of exosmosis, because this salty solution is a hypertonic solution. And the other slice that was in the tap water, in contrast, will actually swell a little lightly, and when you try to break it, it'll feel a little rigid, and it will not break as easily or will not feel as soft, because it has absorbed water, because water in this case is acting as a hypotonic solution, and the cells have swollen, so it has become a little stiff. So that was experiment number 1 for you to perform. Moving on to experiment number 2. For this you need cabbage leaves. You can use the first 2 covering leaves of the cabbage that your mother usually throws off. Second thing that you need is some water containing food color. It is very easily available at home. If you do not have it, use any ink. Any ink from the fountain pen or from old sketch pens. I have used an amber colored food color. Prepare the solution and dip these cabbage leaves as shown in the picture and leave it for a few hours. In second case, what I have done is a red solution, again a food color dissolved in some water, and then I have kept a colocasia leaf. You can see, please look at the leaf carefully how it appears, and we keep it for some time and then observe the changes after a few hours. After a few hours, they appeared like this. The cabbage leaves have absorbed water by the process of osmosis. This colored water, as it reached the parts of the leaf through the veins, you can see the color has also reached those parts. Also, in case of colocasia leaf, you can see the veins are getting red, and you can see the red color on the periphery of the leaf. Moving on, so this is a very simple experiment. You please make sure you perform it. Moving on to osmosis in daily life. Now, osmosis also is a process that happens around us in several instances. Let's discuss a few. Now, we all are fond of eating pickles and jams, and we know how to prepare them. The reason why we can enjoy pickles and jams for a longer period of time without any fear of their spoilage is osmosis. Actually, vegetables and fruits are very prone to bacterial attack because they contain a lot of water, which is the primary requirement for any kind of microbial growth. But when we put these fruits or vegetables in a very high salt solution or high sugar concentrations, this solution becomes hypertonic to bacterial cells. And the bacterial cells, when they enter these solutions, or you can say the pickles or the jams, they lose water, which kills them by dehydration before they can cause any food spoilage. So this is osmosis happening in real life inside your kitchen. Also, you have seen that how plants wilt when you do not water them, but when you water them, they kind of spring back into shape, again by the process of osmosis. You also see osmosis in daily life happening as you see in the picture. What you see here is a person gargling with salty water. Why do you gargle with salty water? When you have sore throat. So when you have sore throat, what is happening inside your throat is that the cells and the tissues around the throat are swollen due to the excess of water. This salt water that we use for gargling is actually has a lower concentration of water than the cells of the throat. So when we are gargling, the water molecules move from the swollen cells of the throat to the salt water, which reduces the pain and the swelling, and it becomes easy for us. So that's why we gargle when we have sore throat. Now, next example is wrinkly fingers. When we sit in the bathtub or submerge our fingers or feet in water for a while or for a little longer, they get wrinkly. This is because of osmosis. What happens is that the skin in our fingers absorbs some water and gets expanded or bloated or a little swollen in regions, leading to the pruned or wrinkled fingers. Let's move on to some experimental activity. Now, this activity you must perform. activity is raisins in hypertonic solution. Now what do you need for this activity is some salt. A few teaspoons of salt will do. You also need some water, which is going to be the solvent. The salt is the solute. You remember what is a solution? You need next is either a few raisins or a few grapes. You can choose whatever is available. What we are going to do now is prepare a solution containing a little concentrated solution containing salt and water. So once you have stirred the solution properly, as you can see in the picture, you're going to put the grape or the raisin in the water. I have put both in two separate containers. Now after a few hours, you can also leave it for a day or two. You will see, as in the picture above on the right side, the raisin has started to shrink because it was placed in hypertonic solution. The salt concentration was more outside, water was less than inside the raisin, so water moved out. It is a classic example of osmosis happening due to hypertonic solution. You can also see the picture below of grape which has shrunken. You can see the wrinkled grape. It was also placed for the same amount of time in the salt solution. Make sure you do this activity and record the observations. Moving on to the next activity, which is raisins in water. Now when raisins are kept under water, what is happening is that the concentration of water surrounding it is more than that of the raisins because the raisins are dry. So water here is acting as a hypotonic solution. Therefore, the water will go inside the raisins and cause them to swell up. In the first solution, in the first picture, you can see it is simple solvent, that is plain water, tap water, and I have put some raisins in it and leave it for a few hours or maybe for a day or two. After that time, what you can see in the picture is that the raisin, there appears a crack in the raisin because it has swollen and it has burst, as we have studied, because of the hypotonic solution. What has happened is more and more of water moved inside the raisin, causing it to swell and eventually burst. And below the swollen raisin, you can see the swollen grape which has actually burst, again due to hypotonic solution. What is the process involved? Osmosis. Moving on to raisins in isotonic solution. These are raisins soaked in some sugar solution. Now what has happened, as you can see, the marked raisins, they are of the same size because they were in the isotonic solution. Now the amount of water going inside in this case is same as the water going out, and hence no net movement of water takes place. Therefore the raisins remain same in size. Now this was the last activity in this video to be performed. Moving on, we have a little fun task for you. Make sure you complete these two tasks. With this, we come to end of this module. Happy learning.